Genevieve gave a long and wonderful introduction. My group has been, a cell division group focused mostly on the cytoskeleton. I'm actually, for a long time, had an aspiration to have some understanding of the evolution of cancer genomes, and because I'm trained originally as an oncologist, but it took me a really long time to kind of crawl down the microtubule and eventually find the chromosome. So I'm going to this will be a little bit more of my personal journey of discovery, a little bit of the chromosome. So the general question that I'm going to try to address today is, how do you get very complex genomes, which is most famously but not exclusively occurring during the evolution of cancer genomes. So that's illustrated over here. This is a particularly extreme example, a HeLa cell that has many tens of thousands of genomic rearrangements and altered copy number and things like that. And so the question is, I want to address how these very complex genomes evolve. And traditionally, we've thought about it in a kind of classic gradual Darwinian mutational accrual model, that mutations happen at a specific rate. Over many cell divisions, you have one after the other piling up and then giving you a complex genome. If you sat back and look at this incredibly crazy messed up genome and just, looked at it de novo, you could start to wonder how long really would that take, and would cancer in fact be a problem in human populations if everything happened gradually and slowly. And so there's been a new idea emerging in the field, which is that complex genomes like this may actually be more parsimoniously explained if we think of them as having evolved through a smaller number of genome catastrophes triggered by cell division errors. And my group has worked on this problem in two aspects. One, which I won't talk about today, is we've worked a lot on the consequences of whole genome duplication, which we showed can drive tumor development, and that's a catastrophic event that triggers a lot of downstream instability. And then I'm going to talk briefly about a phenomenon. I'm going to focus today mainly on a phenomenon that Genevieve alluded to called chromothripsis, and the mechanism of this really interesting, very catastrophic event that can happen to the genome. Okay. So chromothripsis was discovered by cancer genome sequencing by Peter Campbell at the Sanger Institute in a paper described only in 2011. So I think this was one of the most exciting and surprising findings from cancer genome sequencing. And interestingly, it's a major mutational process that was only discovered very recently. But the mechanism underlying wasn't known. So chromothripsis has two interesting aspects. The first is that it's typically confined to only one or a few chromosomes, and it looks like from the sequence that somebody would take these, small number of chromosomes and completely fragment them, and then reassemble the fragments in random order and orientation to generate a completely novel chromosome. So there are two questions. Why is it highly localized? And I'll show you in a minute that it has a very unusual DNA copy number profile that suggested that it might be some kind of all-at-once catastrophe. And so the phenomenon was very interesting. The mechanism wasn't known. And in this pair of papers here, we proposed and I think made initial steps to validate a mechanism driving the formation of chromothripsis, which involves a very interesting general problem that I know is of interest here, which is the abnormal nuclear structure in cancer, known as nuclear atypia. This is a DNA stain of a rather extreme example. And we particularly focused on one specific kind of abnormal nuclear structure that's very common in cancer, called micronuclei. I'll show you a little bit about their origin later. But we had the hypothesis that the physical isolation of one or a few chromosomes in an abnormal nuclear structure could actually generate this phenomenon called chromothripsis because of DNA breakage occurring in these micronuclei. And we were able in the 2015 paper to basically recreate chromothripsis in the laboratory by generating micronuclei, watching aspects of their behavior, and then isolating the daughter cells from a micronucleated mother cell and doing single-cell genome sequencing to demonstrate that this caused chromothripsis, giving us a very nice system now to understand the underlying mechanisms. So today I'm going to tell you, hopefully if I have time, 2 unpublished stories about the mechanism of chromothripsis, one involving the nuclear envelope and the other actually addressing the DNA fragmentation mechanism. Chromothripsis was initially thought to be rather rare because very low sensitivity array-based technologies were used to study it. And now with the advent of many studies using extensive whole genome sequencing, we know that chromothripsis is actually a major mechanism shaping the architecture of human cancer genomes. So chromothripsis is extremely common. For example, in this paper on human pancreatic cancer, they report that chromothripsis occurs in 65% of these tumors, with typically multiple chromothripsis events explaining more than half of the genomic rearrangements in pancreatic cancer. So this is a good example, like whole genome duplication, of a catastrophic event that has a major impact on the structure of cancer genomes. I should mention that chromothripsis is in no way specific to cancer. It actually causes certain human congenital diseases. And it's no way specific to humans. It can be observed in plants in very interesting contexts, for example, during models of interspecies hybridization. So this is a general mutational process that in and of its own right, I would like to think, irrespective of the translational implications, is interesting to understand. Okay, so then just a quick, so this is an example of a chromosome that's undergone chromothripsis to familiarize you with the features. The links here show the rearrangements of segments of the genome that shouldn't be linked to each other, but that are. And then I want to emphasize here the unusual copy number pattern. So if you pretended you were like a little person walking from one telomere to the next and walking along the chromosome and counting how much DNA was below your feet, what you would find in this particular example would be 2 copies of the chromosome with heterozygosity, because you have 1 copy from mom and 1 from dad, going down to one copy or with hemizygosity, 2 copies, 1 copy, 2 copies, 1 copy. So this was really a completely bizarre and unexpected pattern. It hadn't been described in cancer before, and there was a sense that this was really what was giving us a clue that this was a novel mutational process. Okay, so I'm only, because I want to focus on unpublished work, I'm going to just show a little animation that summarizes this published work, and then I'm going to get directly into new stories. Okay, so this is saying that micronuclei can originate in a variety of ways. You can have an abnormal mitosis, for example, if you have extracentrosomes. I see Michelle in the audience here, which can set up bad mitoses, which can cause chromosomes to get caught in tug of wars, lag, and then be left out in micronuclei when the nuclear envelope reassembles. I think an important point to realize is that a single double-strand break that persists in mitosis does the very same thing. So a double-strand break can have multiple downstream consequences. These micronuclei we show undergo very poor DNA replication. As shown by Emily Hatch and Martin Hetzer, the envelopes are fragile, spontaneously rupture, and by interesting and mysterious mechanisms, they, upon exposure to the cytoplasm, the chromosomes are damaged, fragmented, and then we showed by single-cell sequencing you can get some fragments going to one daughter, others going to the other. Once they're incorporated into a daughter cell nucleus, Now they get stitched together, presumably by end joining mechanisms. And the fact that you have some bits that are present in one daughter and missing in the other then generate this, perfectly recapitulate this oscillating DNA copy number. So the fragments that are lost are not really lost. They presumably just went into, if this cell developed into the cancer cell and this cell died, then you would get the apparent loss, but it's not really loss of DNA segments. Okay, so if we then were to summarize, you would say that the mechanism of chromothripsis involves the generation of a micronucleus. And then for some reason, the nuclear envelopes on these structures are fragile and undergo spontaneous rupture. And then for what we think of complex and interesting process, they are then fragmented and then stitched together to make chromothripsis. So the first half of my talk, I'm going to address the issue of why the membrane on these structures are fragile and rupture. And then in the second, I'll talk about incomplete and provisional understanding about why the chromosomes are then fragmented. Okay. To understand, addressing the question of why the nuclear envelope on micronuclei is fragile addresses, I think, a generally interesting question in cell biology, which is there, and if so, how, is the completion of chromosome segregation coordinated with the assembly of the nuclear envelope? Okay, so the question is, during mitotic exit in metazoan cells, is there any kind of checkpoint mechanism that monitors chromosome position? Is there any kind of active coordination? Or is it like a free-for-all, so that you segregate the chromosomes, the nuclear envelope reassembles, and whatever it is, you get what you get? So we're going to address that question. So in thinking about how you might have a problem with the nuclear envelope on micronuclei, you have to start thinking about nuclear envelope assembly in general. So I'm going to show you a couple slides on sort of my take on an interesting, complex, and rapidly changing literature. So don't take everything I say completely at face value. I'm just trying to give you my sense of the current state of this interesting field. So when you look at a nucleus and interphase, you normally think of that as a rather homogeneous structure with nuclear pores distributed all around it. But in fact, transiently, in telophase, when the nuclear envelope is assembling, there's an interesting phenomenon. So what happens is that you very rapidly get membranes binding the decondensing chromosomes that are chock-filled with nuclear pores. And this is referred to as late mitotic nuclear pore assembly or post-mitotic nuclear pore assembly. But interestingly, the nuclear pores and other set of proteins that I'll refer to here as non-core proteins, all are initially very restricted to the outer surface of the decondensing chromosomes. In here, not shown, are dense bundles of a telophase spindle, and adjacent to this region where the spindle is, you get a concentration of another group of proteins, which I'll refer to as the core proteins. I don't want you to remember all the names. I do want you to remember that one protein we're going to look at a lot is a membrane protein called emerin. So basically, transiently, you have two teams of proteins that then assemble, and then within 10 minutes, that's kind of homogenized, and I'm going to show you a little movie that illustrates that. And then the first thing that we did is just ask whether chromosomes that lag within the spindle undergo the same temporospatial pattern of nuclear envelope assembly. Okay, so this is just my take on normal nuclear envelope assembly. Again, here's the spindle. You complete chromosome segregation. The chromosomes start decondensing. There's some interesting points about this. You have this ER membrane with the nuclear pores in green here, and you have the core domain, which is initially homogeneous, but then concentrates by the spindle here. And then something interesting happens. So in interphase, these core domains lacking nuclear pores kind of break up by a mechanism that's not so clear. And then in a process that requires the assembly of new nuclear pores that requires transport through the old ones, you then start populating the interphase nucleus with more nuclear pores. So in a G2 interphase nucleus, you have the nuclear pores, half of them were assembled in 10 minutes in telophase, and the other half slowly occur by this interphase pathway. And because you require transport, the interphase pathway would depend on the success of the post-mitotic nuclear pore assembly. Those are points. Okay, so I wouldn't have gone through this long rigmarole if there wasn't a big effect, which is that if we look at the lagging chromosomes here shown in DAPI, you can see that the core protein emerin assembles very robustly on lagging chromosomes, but nuclear pore proteins, for example, this Y complex protein, are virtually missing. That's also true of other things like the lamin B receptor that are correlated with the nuclear pore proteins. This effect is not subtle. In 3 different cell lines using multiple markers, you see robust assembly of the core group of proteins, and then you're missing the non-core group of proteins. Somewhat more variably, apparently with some partial correction, in interphase, these defects persist. You see robustly the core proteins on the micronuclei that result from the missegregation, and lamin B receptor is particularly dramatic in being missing, but nuclear pore proteins are also more variably missing. There's been some disagreement about whether these micronuclei assemble nuclear envelopes and import normally. That was the proposal from Martin Hentzer's group. But I'm not going to show you all of our data. We very carefully looked now at nuclear import and measured it in good quantitative terms, and I'll just tell you that there's consistent with this dramatic nuclear pore assembly defect, there is also a very significant defect in nuclear import, and many key proteins that are required for DNA replication and nuclear envelope integrity, like B-type lamins, fail to normally accumulate in these micronuclear structures. So that's all very interesting, but that raises the important question of what's going on and why do you have this assembly defect? So as we first started thinking about this, we went through the literature and found this interesting paper from Elder Mayato's group. What Elder had proposed was that that during mitotic exit in metazoan cells, there's very careful checking of the position of chromosomes, and that cells use the phosphorylation gradient from the mitotic kinase Aurora B that localizes in the middle of the spindle and then spreads its phosphorylation gradient towards the poles of the mitotic spindle, that the cells are using this gradient to establish the position of lagging chromosomes and then actively preventing the assembly of nuclear envelope on these lagging chromosomes, essentially giving them time to catch up and be integrated into the main chromosome mass, ensuring that during cell division you get one and only one nucleus. So this was a really interesting proposal. However, we had several pieces of data that started to make us wonder about this model. First, if we just used Aurora B inhibition, we could repeat that inhibiting Aurora B would reverse some of the assembly defect of the nuclear pore complexes, but we got a weird result. The recovery happened in 50% of the anaphase cells, but not in the other 50%. So that was quite curious. And then we did experiments like this where we inhibited the motor that walks Aurora B from the main chromosome mass out into the middle of the spindle. That's shown here. So if you knock down that motor, you can abolish the mid-zone localization of Aurora B, and in data that I'm not going to show you here, we can see that you completely flatten the Aurora B gradient. And when we do this, now we still are not recovering the nuclear pore proteins on the lagging chromosomes. So all of these experiments and others that I won't have time to discuss made us wonder, could there be something else going on? Could the Aurora B inhibitor maybe be acting differently, indirectly? And the thing that we started wondering about were my old friends, the microtubules of the mitotic spindle. Because there are papers that have suggested that Aurora B has a negative impact on nuclear envelope assembly and nuclear pore assembly, although the mechanism isn't really well understood. But the most famous thing that Aurora B is known to do is to regulate the stability of microtubules. So in anaphase, if you inhibit Aurora B, it was very well established from this series of papers that if you inhibit Aurora B, you take an anaphase spindle from this nice bundled, robust structure like this to this weak and largely unbundled structure. So what's happened here is that when you inhibit Aurora B, you activate enzymes that depolymerize microtubules, like MCAC for the cognoscenti here. So you get a huge effect on the spindle with Aurora B inhibition. And in fact, if you go through the old literature, there's actually quite a bit of data suggesting that microtubules somehow are inhibiting nuclear pore complex assembly on chromosomes. For example, there was a very beautiful study from Hiro Funabiki used in Xenopus egg extracts in vitro, where he showed that if you surround chromosomes with polymerized microtubules, something that happened in depletion of a particular protein, that you could block nuclear pore assembly and that you could reverse that, for example, by treating the extracts with nicotazole. So we started wondering about whether the previously reported Aurora B effects could all be explained by indirect effects on microtubules. And so we designed an experiment shown here. I'm not going to have time to show you every experiment that we've done, but we designed an experiment to test the idea that this 50-50 effect of Aurora B inhibition could be explained by a narrow time window at which depolymerizing microtubules might be important to inhibit nuclear pore complex assembly. So the experiment was done as followed. We synchronized cells in G2, and then with a CDK1 inhibitor, and then we released those cells in the presence of a drug that induces chromosome missegregation, an MPS1 inhibitor. So we made lagging chromosomes, and then we, at the approximate time of starting anaphase, we added either the Aurora B inhibitor or what's shown here is nicotazole to depolymerize the microtubules. The important point in this experiment is that synchrony in none of these experiments is perfect. So the moment of drug addition, you have a mix of cells. So we were able to find cells, because we were doing live cell imaging, where we hit the cells with the drug at every point from the start of metaphase all the way through the end of telophase. And then we could assay whether microtubule depolymerization made any difference, whether it happens early in mitosis or later in mitosis. So here's the control. Here's a lagging chromosome. You can see the dense bundles of telophase spindles here. You can see that there's very little nuclear pore complex protein on this lagging chromosome, a little dot here. And you can see that the lagging chromosome is basically completely ensheathed in a bundle of microtubules. Now, if we go early in anaphase, 4 minutes into anaphase, depolymerize the microtubules. You can see the microtubules are gone here. Here's the lagging chromosome. Now you can see that we can, in early anaphase, recover nuclear pore complex proteins on the lagging chromosomes. But interestingly, and not in agreement with the checkpoint model, this nuclear pore complex assembly is incredibly heterogeneous. And in fact, where we see the densest bundles of microtubules, we're typically missing nuclear pore complexes. If we now come back just 2 minutes later in anaphase and depolymerize the microtubules, we get a very different effect. You've completely lost the ability to reverse the nuclear pore complex assembly. So there's a couple of points that are interesting here. The data are consistent with microtubules inhibiting nuclear pore complex assembly, but it suggests that somewhere between this time interval, something irreversible has occurred and makes the nuclear envelope defect on the forming micronucleus permanent. So we wondered what could cause, so this is very different from a checkpoint effect that would be beneficial to the cell. This would say that there's a nuclear envelope assembly defect that's not only bad, but that it's also irreversible. So what would cause that irreversibility? And again, this whole project was a bit like a treasure hunt through old literature for us. And as you go back and read the nuclear envelope literature, it actually makes quite a bit of sense. So what you can see here is that on these lagging chromosomes, this is a super resolution image, you can see that this membrane protein emerin is completely ensheathing the lagging chromosome, but you're missing the nuclear pore complex proteins. So what it looks like is that the chromosome actually gets physically wrapped in a membrane that lacks nuclear pore complexes. And that in itself could, in fact, be the irreversible event. And in fact, if you go back and look at these old papers, for example, that paper from Ian Matai, you see exactly that. So in that paper where Toby Walter, as a graduate student, was working on nuclear envelope assembly, what he did was deplete the extracts of a key nuclear pore complex protein. And then if you do that, the nuclear envelope assembles in a nice double membrane, but it's completely depleted of nuclear pores. If you then try to add back the nuclear pore complex to complement the extract, if you do that early, just like in our experiment with the nicotazole depolymerization, you can recover the nuclear pores onto that forming membrane. But once the chromosomes have become wrapped in a double membrane that's depleted of nuclear pores, it's irreversible and you cannot do the add back. So we think that's very likely the explanation. This is with Alexei Kochikov. We've been doing some correlative light and electron microscopy experiments. And what you can see here is, so here what Alexei's done is to time the constriction of the cytokinetic apparatus here with timing into telophase here. So that gives you a nice line, so you can use the length of the constriction as a way to time by Clem where the cell was at the time in which you did the fixation here. And what you can see, this is really a remarkable result as far as we're concerned. So what you'll see is that at the onset of telophase, you initially, none of the chromosomes, the main chromosomes or the lagging chromosomes, none of these chromosomes have nuclear envelope assembling on them. These are different Clem experiments done in red here with no nuclear envelope. And then just a minute or two later in telophase, what you'll see is that all the chromosomes, including on the main chromosome mass and the lagging chromosomes, now assemble nuclear envelope. But by EM, we can confirm that it's only the ones on the main chromosomes that get nuclear pores, and the ones on the lagging chromosomes are deficient. So that's shown here on the main chromosome mass. I hope you can see it. There's a double membrane here. You can see little constrictions where you have the nuclear pores here. Here's an en face view where you can see one nuclear pore. And then on the micronuclei, you get a beautiful double membrane here, but it's devoid of nuclear pore complexes. Okay, so we think that the microtubules are inhibiting nuclear envelope assembly, at least with nuclear envelope with nuclear pores, but that you can get an envelope lacking the pores, and once you have that, it's irreversible. If you really believed all that, one prediction of that model would be, if the spindle is really causing all the problems, is that if you were able to find a trick to be able to take a missegregated chromosome and move it outside of the spindle into the periphery of the mitotic cytoplasm, the prediction would be that you should reverse the defect in assembling nuclear pores, you should reverse the defect in the import, and then you should also correct the DNA damage and nuclear rupture of these micronuclei. And my very clever graduate student, Shiwei Liu, thought about how to do this, and one of the ways he did it was by so motor proteins start at the poles of the mitotic spindle, and they use the asymmetric positioning of post-translational modifications of tubule to guide the congression of chromosomes into the middle of the cell and not out to the periphery. So you have more detyrosinated microtubules in the spindle, which the SEMP motor likes to walk on, as shown by Elder Mayato. And it prefers less to walk on the astral microtubules. If we knock down an enzyme that then homogenizes this so that everybody has the detyrosinated microtubules, the motors get confused, and about half the time they'll walk the lagging chromosomes into the middle of the spindle, but half the times they'll also walk them out to the periphery of the cell. And in this way, we could get mixed populations of cells that have missegregated chromosomes in the spindle or out in the periphery of the cell. And this is an experiment that we've done in many different ways in a couple different cell lines. I'm just going to show you one live cell imaging experiment. What you're looking at here is a cell that's you're going to see a slice of a cell in metaphase over here. So here's the main chromosome mass here, two peripheral missegregated chromosomes here. So the chromosomes are in red and nuclear pore complex proteins are in green. You can see the cells in metaphase here. Anaphase onset starts here. The main chromosome masses are splitting. As the main chromosome masses split, you can see chromosomes that are lagging in the middle of the spindle. And then what you can see quite dramatically is that the peripheral chromosomes assemble nuclear pore complex proteins very beautifully, but as expected from what I just showed you, all the chromosomes that lag in the middle of the spindle do not. So peripheral localization of chromosomes restores nuclear complex protein assembly. If we then do live cell imaging of these micronuclei that we know are derived just from the peripheral lagging chromosomes, what you can see is that you massively suppress the frequency of rupture. So of the ones in the spindle shown here in blue, something like 60% will rupture in the next 18 hours during interphase, but the peripheral ones are dramatically suppressed. Only about half of the micronuclei that rupture actually undergo DNA damage, which is why you have less damaged ones than ruptured ones here. But comparing the peripheral and the central ones, again, we can see a strong and statistically significant, although not perfect, rescue of the DNA damage in these structures. Okay. Okay, so to summarize the first part of this talk, we think that this is starting to elucidate an important step in the mechanism of chromothripsis, showing the mechanism of nuclear envelope breakage. So here's a cell now in late anaphase. You can see no nuclear envelope is assembled, but we hypothesize that you can get the core proteins on both the lagging chromosomes and the main chromosomes, but only the main chromosomes can recruit nuclear pore complexes. And we think that this is because of inhibition by spindle microtubules. The mechanism of this obviously is very interesting and needs to be determined. But it could be, Jan Ellenberg, for example, has a beautiful recent paper suggesting that this late mitotic nuclear pore assembly occurs because you have pore precursors assembling in fenestrated sheets of ER that then wrap the chromosomes. So you could imagine something ridiculously simple where the reason that you don't get the nuclear pores in the central portion of the spindle is that those ER sheets are not able to access the lagging chromosomes. And then you get the concentration of the core proteins here. You wind up with an interphase cell that's markedly deficient in nuclear pores, and then you get nuclear rupture, chromosome fragmentation, and chromothripsis. We would propose then that in mitotic exit in mammalian cells, I would propose is the most vulnerable part of the metazoan cell cycle. And that this is explaining why chromosome breakage-fusion-bridge cycles, chromothripsis, are all originating from errors in this step of the cell cycle. And that's because nuclear envelope assembly is only loosely regulated, being dependent on the timely disassembly of the mitotic spindle, but there is no, in our view, strong checkpoint that corrects errors at this point. And we think the lack of these precise controls is exactly why you're making a lot of genomic rearrangements here. Okay, I have to apologize that I went a little bit slow. How much time do we have? We got started a little bit late, but so I'd like to address our baby steps at trying to understand the mechanism that causes chromosome fragmentation. We're working hard on trying to understand why, when the envelope breaks on micronuclei, you get DNA damage. I'm not going to report progress on that, but in we think that once those chromosomes are in the cytoplasm, in mitosis, they're undergoing a whole nother round of DNA damage. And I'm going to discuss work here on trying to understand why replicating DNA in mitosis might be very bad and could be an origin of chromosome fragmentation that leads to chromothripsis. Okay, so the idea that attempting to replicate DNA in mitosis could be a bad thing goes all the way back to early experiments with micronuclei and early cell fusion experiments from Johnson and Rao, where in the Johnson and Rao experiments, you can fuse an S-phase cell with a mitotic cell, and you get this, what was described as a pulverized appearance of chromosomes. This was itself, in the early cell fusion days, highly controversial. So initially they called it pulverization, and they assumed that it was chromosome fragmentation. But then most of the field came to exactly the opposite conclusion, that this was merely discontinuous condensation of DNA with nice little bits of intact replicating DNA connecting the decondensed bit. So this is why the idea that mitotic replication might be a bad thing kind of went silent in the field, and people kind of ignored it for a long time. But subsequently, there have been other examples, like the expression of common fragile sites, so difficult to replicate regions of DNA are now known from Michel Debattis and many others to undergo fragmentation, to undergo breakage during mitosis. And then micronuclei, we got interested in this, of course, because we're sort of micronuclei focused, because micronuclei, they replicate DNA poorly, but they keep trying. So they're still replicating in G2 as shown here. And I'm going to show you in a second, these G2 replicating micronuclei have very little evidence of DNA damage. But once they get into mitosis, they're still replicating their DNA, and they have evidence of we think clear evidence we've shown of chromosome fragmentation. And now I'll show you they start to get extensive DNA damage. Okay, so I just told you everything on this slide. So here's the G2 cell. So again, this is our favorite. We're doing live cell imaging and then fix the cells and then same cell, fixed cell imaging. This was everything on the nuclear pore project was done in this way, and a lot of our work here is done that way. So here's a micronucleus, and you can see that in G2, when the micronucleus is intact, I'm not showing you the markers of that, but take my word for it. You don't have a lot of single-stranded DNA marked by the localization of the single-strand DNA binding protein called RPA, and you don't have a lot of gamma H2AX indicating DNA breakage and damage. But if you then take these very same cells and release them into mitosis, you get massive accumulation of RPA1, so lots of single-stranded DNA, and you get a big, gigantic blob of DNA damage coming from H2AX. And I'm not going to show you this, and it's still a work in progress to get the numbers up, but in the 6 or 7 examples where we've done whole genome sequencing on these G2 cells with intact micronuclei, we see no evidence of chromothripsis. But if we now let these cells pass into mitosis and recover the 2 daughter cells and sequence them, what you can see, here's daughter 1, you can see massive chromothripsis on 1 chromosome here. Actually, it's a little light here. There's some fragments here that make chromothripsis, and the rest of the genomes are silent. So passing through mitosis with a chromosome from a micronucleus is not good. And it's a source of generating chromothripsis, and we'd like to understand the mechanism. Now, we're working on this in cell systems, I'm not going, that work is all in progress, and I'm not going to discuss it because it's not yet terribly conclusive. But one of the things that's held us back in understanding DNA replication and mitosis is that mitosis is short. It only lasts like 50 minutes or so in the average tissue culture cell. So if you want to make a genetic manipulation to ask whether this, that, or the other protein is required for replication and or damage in mitosis, you have a very narrow time window in which to do your experiment. Furthermore, many of the proteins that you might want to study have key roles in normal S phase. So if you're knocking down some protein for 24 hours and it's messing up S phase and you see some damage in mitosis, it becomes very difficult to say that the damage was really because of something that happened in mitosis rather than earlier in the prior S phase. So to address this problem, I hooked up with a good friend and now close collaborator at Harvard Medical School, Johannes Walter. And what Johannes did when he was a postdoc with John Newport was to develop an elegant system to study mitotic DNA, to study replication in Xenopus egg extracts. So, and important, this is cell-free and nucleus-free. So it's one extract in which you license replication origins and load the MCM complex, and then another extract will fire them and give you complete replication. And so I suggested this experiment to Johannes, which is just to add mitotic cyclins and drive the extracts into mitosis, and asked what happened. There's a lot I won't have time to show you. We can show that we get normal chromosome condensation, recruitment of condensins. So it's hard to say that it's exactly mitosis, but I would say that these extracts are now very mitosis-like. Okay. So the way this works, you take a plasmid and you throw it into the extracts, and then you add hot nucleosides. And what you can see is in the normal S-phase extract is that you'll see some replication intermediates when you run the products out on a gel, which then rapidly are converted into open-circled, supercoiled, full-length plasmid replication. So that was good. So next thing we did was to throw in the mitotic cyclins, and we got a very disappointing result to me, which was that replication starts a little earlier and goes faster. That's expected because CDK activity activates the firing of replication origins. But we saw nice normal products and didn't see anything terribly abnormal. So I was depressed about this, and then, but my Johannes gently reminded me that he published exactly this result in 2013, where he added cyclin A and cyclin B, and he said, aha, here I told you this was going to be the answer. But I wasn't ready to give up, because really the relevant substrate in mitosis should be a stalled or collapsed DNA replication fork. Because that's what would happen at a fragile site where you haven't finished replication, right? You would have regions of replicated DNA joined up to unreplicated segments, and because of poor replication in micronuclei, you get the very same thing. And so what we did here is to take the plasmid that had many copies of the operator sequence for lac repressor, and now replicate the plasmid in the presence of the lac repressor, which creates a protein barrier that stalls the replication forks very uniformly. So the circle plasmid then becomes a kind of theta structure here with unreplicated segments here and replicated segments here. So that's shown here in the S-phase extract. You can see that everything is very efficiently converted into the theta structure. Now, something exciting happened when we add mitotic cyclin, which was that these replication forks are apparently very rapidly being broken. You see a big smear here. And then what happens is that all of the broken fragments seem to get ligated to each themselves into a kind of big snarl that won't even migrate into the gel very well, which is the well product up here. Okay, so replication in mitosis conclusively in this experiment is definitely bad and leads to the breakage of the forks and aberrant ligation of the broken products. So the next thing we wanted to know is what's going on at these forks. And what we did is that we took our plasmid and we digested it with an enzyme. So if you take the theta structure and cut it once, since the cutting site gets replicated, you wind up with a so-called double Y structure. You can see that here. That goes from here to here, joined around here. Hopefully you can see that. Okay, so that's the double Y structure here. And in the S-phase extract, you can see that slowly it gets bigger because very slowly the lac repressors are not a perfect barrier to replication, and the replicon slowly chugs through, and that's why it gets a little bigger here. Here we add mitotic cyclin, and you can see that we get the collapse of the replication forks, the breakage of the forks. And then you can clearly see some abnormally small products here. There's probably other stuff abnormal that's smeared up in the gel here, but we could see these are clearly abnormal, and we cut them out of the gel, transformed E. coli, and then sequenced them to get a sense of what's going on. There's a lot to unpack in this experiment. For Elaine, I'll say that we got a lot of products that were the result of a process called single-strand annealing. I'm not going to talk about that too much because we have a lot of repeats in this single-strand annealing. Joining mechanism is kind of specific for having long track repeats. But what I will talk about is that many of the junctions showed extremely complex genomic rearrangements with evidence of the possibility that you had DNA replication template switching. Okay, so this is an example showing what's happened with one of the rearrangements where we have 5 copies from one side and 15 copies from the other side of the fork. You can see that we got breakage of the fork here, annealing of the template behind. There are 3 copies of unique space. There are 4 unique spacers within the lac repeats, and then there are 48 copies of the lac repeats. So they come in sets of 4. So we can see here that we had part of the operator that's fused to the 3rd spacer, and then that extended, and then we got to jump back to right behind the 1st spacer sequence over here, which then extended and gave us complex products like this. So this is obviously an inference, but it looked very much like we were getting switching of the templates of replication. So not only are you getting breakage of the forks, but the replication that's repairing it is defective and making errors jumping around the plasmid. That was very exciting to us because when chromothripsis was first discovered, there was a raging debate about whether it was caused by fragmentation and religation of the chromosomes or from this type of replication template switching error where you had a replication fork that stalled, jumped behind the blue segment, jumped over to the green segment, et cetera, et cetera. So this has been documented most beautifully by James Lupsky and Frank Ira with many contributions for a lot of people in the field. You can definitely get this kind of template switching replication. In our study here, we actually saw some evidence for small-scale template switches at the rearrangement junctions. For time, I won't take you through the evidence. But we would propose that these two models are actually not exclusive, and maybe the same initiating event, like making an abnormal nuclear structure, is causing both bad replication and chromosome fragmentation and religation. This kind of small-scale template switching looks a lot like a process called alternative non-homologous end joining, which in human cells is largely mediated by a repair polymerase called DNA polymerase theta. No connection to the theta structures I showed you before. I'm just going to summarize this without going through this slide, that if we deplete Pol theta from the extracts, we significantly reduce the numbers of aberrant replication products, and we also suppress the amount of replicated DNA. And we interpret that to mean that in the absence of Pol theta, you can't join the fragments. If you don't join the fragments, they get degraded in the extracts. So you get some aberrant junctions and some degradation, which explains the phenotype that we have here. So I just have one more piece of data to show you, which is, the really interesting question is what causes the replication forks to break in the first place? And we started wondering, so here's a kind of cartoon of a stalled replication fork. You have the polymerase is back here, and out in front is the replicative helicase, the MCM 2 through 7 complex. When it's activated, it's called the CMG complex. And this stalls here at unreplicated DNA. Interestingly, in a bunch of beautiful, elegant experiments from Johannes's lab, in conditions where you need to make a nick in replicating DNA to fix a break, as occurs in the Fanconi anemia repair pathway, you absolutely need to get rid of the MCM complexes in order to make the incisions that allow the initiation of repair. So that's a beneficial, controlled way to make incisions, but we wondered whether the breakage that happens in mitosis could be a completely uncontrolled massive breakage that requires the removal of CMG. Because at the end of the cell cycle, the cell needs to get rid of all of the replosome proteins from the chromosomes in order to reset the whole program to allow the next program of DNA replication. So here's an experiment. What we've done here is we replicate the plasmid here with the replication stall, with or without mitotic cyclins. And then what we do is to pull down the plasmid, because we have biotin on the lac repressors, and then we can blot for proteins. So what you can see here is that in the S-phase extract, you have lots of MCM complexes here, as expected. But in the mitotic extract, as I suggested to you, you clear off all of the MCM complex and you unload them all. Okay? This requires the ubiquitination of the MCM7 subunit. So here, MCM7, you can see, runs as a smear. We can collapse that ubiquitinated smear into a single band by adding a nonspecific deubiquitinating enzyme. Okay? And then the really interesting result, this is the last result I'll have for you, is that if we block the unloading of MCMs that are ubiquitinated involves a protein translocase called P97 that disassembles many complexes in the cell and will pull MCM out of chromatin. So there's an inhibitor of MCMs, of the P97. If we add that to an S-phase extract, nothing particular happens. So P97 doesn't do much for normal DNA replication. However, if we, here's the control mitotic extract where you can see the fork collapse and the aberrant replication products. If we now inhibit P97 and we prevent the extraction of MCM from the chromosome, and that's shown over here. I forgot to mention that on the pull down. If we inhibit P97, we lock MCMs down onto the chromatin. What you can see here is that we completely restore normal replication. So we force MCMs to stay on the plasmid when they'd normally be unloaded in mitosis, and the forks don't break and replication continues. Okay, so just a couple models. So we'd like to propose that you can have obstacles that stall the replication fork and might make these stalled forks persist into mitosis. In mitosis, then, you have a normal program that's trying to reset the cell cycle, and it's ubiquitolating MCMs and pulling the MCM complex off of the chromosome, and that's done by the P97 translocase. That triggers the unloading of MCMs and creates a little bit of naked single-stranded DNA that would be an appetizing substrate for nucleases like MUS81 and Gen1 that are known to be activated in mitosis. That causes DNA breaks, and then you could get Pol theta-mediated end joining or various processes that would then give you a mechanism that would go from mitotic replication into chromosome rearrangements, chromothripsis, rearrangements in micronuclei, fragile site expression. Obviously, now that we know more about this process from studying it mechanistically in an in vitro system, we're very excited to do more experiments now in cells. I just finally want to end on one little model. So if you have an unreplicated segment of DNA that persists in mitosis, it's been known from Steve West and others that nucleases will then break that unreplicated segment of DNA. And the thinking is that this breakage of chromosomes would somehow be beneficial to the cell. But it's always been kind of puzzling to me why this would be so beneficial, because here you have an unreplicated segment. If you cut here and here, you're going to get highly abnormal products out, right? You'll take the ends of the chromosomes and fuse them, make these kind of isochromosomes. You'll break the chromosome, you'll generate acentric chromosome fragments. The benefit of the cell, you could say this is sort of like a Hail Mary pass, and it's the cell's last chance to do anything to separate these entangled sister chromatids before you absolutely have to divide. But what I'd like to propose is that if the breaks are actually targeted to these sites where the leading strand template is, where MCMs are, what you would effectively do is make a little clip here, a little clip here, then you have an intact chromosome here, which could be joined up to a chromosome. You'll make a little deletion here, but you'll preserve chromosome segregation and at least promote to some degree the fidelity of transmitting the genome. So obviously we haven't demonstrated this, but I'd like to propose that this might be one interesting logic behind why the cell goes through this various gymnastics to do this. Okay, and then I'll just stop here. The early work on chromothripsis was driven by two very talented postdocs. Chengzong Zhang is now an assistant professor in the new computational biology department at Harvard. Alex Spector is an assistant professor at radiation oncology at Dana-Farber and Harvard Medical School. That project was a collaboration with Matthew Meyerson. The work on nuclear envelope assembly was done largely through a collaboration between a super talented graduate student, Shiwei Liu, with a postdoc, Minjung Kwan, and then I showed you collaborative work with Alexei on this. And then the experiments I showed you were all done by one postdoc who's jointly supervised by myself and Johannes Walter Lindeng. Okay, thank you for your attention.